James is Professor of Cancer Genetics at Newcastle Centre for Cancer. He has a specialist programme funding from Blood Cancer UK, which funds his work to develop novel treatments for leukemia and to identify the underlying genetic causes of leukemia, including CLL. James, can I hand over to you now, please?
Yes, thank you very much, Brian, and thank you for inviting me to speak today. So my background is quite different from David's. I'm not a medic, I'm a research scientist, so I run a research group at Newcastle University. And I've worked on cancer genetics for all of my career, and for the last 10 years or so on chronic lymphocytic leukemia. So, but prior to that, I've worked also on numerous other human cancers, including colon cancer, breast cancer, acute myeloid leukemia. We do quite a lot of work on as well. So I have very broad experience in cancer genetics. So what I thought it would be quite useful to do would be just to have a chat about human genetics in general and just describe some of the types of variation that we see in humans and how these might contribute to risk of developing cancer in the first place, and also how genetics might then impact on disease progression and ultimately on survival. Oops. So we're going to be talking about this fabulous molecule. This is DNA. Dave mentioned chromosomes in his talk. So chromosomes are made up of this wonderful molecule called DNA. And I've shown here a picture of a cell. This is a single human cell, and the blue bit in the middle is the nucleus, which contains the chromosomes and the DNA. And this is what the molecule looks like. The structure of DNA was discovered by these two chaps, a Brit and an American. So you can see Francis Crick is the British scientist on the right, and James Watson was the American person that he worked with on this. And that's their original DNA model that they made when they discovered the structure in the early 1950s, and something for which they received the Nobel Prize for. When I first started working at Newcastle about 15 years ago, there used to be a model of DNA made out of shopping trolleys very close to Newcastle Central Railway Station. But unfortunately, I'm disappointed to tell you that it's now been taken down. But that used to excite me, going to work every day, working on DNA to see the shopping trolley structure. So DNA is made up of things called bases, or essentially letters. And there's quite a lot of them in the human genome. There's 3.2 billion of these letters, but in fact there are only 4 types: A, T, C, and G. So that's it, essentially 4 types of letter, but 3.2 billion of these in the human genome. And for most of us, that sequence is identical. So most of those letters are identical between any 2 individuals. But there are approximately about 100 million differences in those bases. That sounds like a big number, but in terms of the proportion of the total amount of DNA in humans, it's actually a relatively small number. So for most of us, we're actually identical in terms of our genetic sequence, other than these differences here that we see. So an important thing to consider as we develop and age is that cells need to replicate themselves. So cells divide. So a single cell becomes 2 cells. And I've shown how that can happen here. Here's the single cell I showed you earlier. And when the cell decides to divide, it first replicates its DNA. And you can see the DNA here in blue. Once the DNA is replicated, it's made a copy of itself. The 2 copies will separate, and you can see that happening here in the top right. So you've got 1 copy at the bottom here and 1 copy at the top. They get pulled apart essentially. And then eventually what happens is the cell will split down the middle to make 2 new cells. Now, this is actually a fundamental process to why we as a species develop cancer. And it's a process that goes on constantly throughout our lives all the time. And in fact, it probably happens much more than you think. So, for example, just in the last 60 seconds, your body has made 300 million new red blood cells. So all of those have arisen through DNA replication and cell division. and about 40,000 new skin cells, again, just in the last 60 seconds. So this process of cell division and DNA replication is going on constantly, and it's happening all the time. So why am I telling you about this? Why is it important? Well, it's important because DNA replication isn't perfect. We would like it to be, but it isn't. And whenever a cell replicates its DNA, unfortunately, this leads to small changes in the sequence. So as well as those differences in the sequence that we carry when we're born that are different between individuals, as we grow old and our cells divide, the DNA acquires changes. These are called mutations. And in genetic terms, we call them somatic mutations, somatic changes. And this is one of the reasons why we actually change physically as we age, because we acquire these changes in our DNA structure as we get older. And this poor chap here has been tested by his wife because he doesn't look how he did when they got married 30 years previously. So I'm just going to bring these two features together into this figure just to illustrate how they interact. So we all start life as one of these. This is a fertilized zygote, human zygote, and this obviously develops at the point of conception. And I mentioned the genome is 3.2 billion bases in every cell, in every human, and about 100 million of these differ between any two individuals. And it's these actually that explain why we look different to each other. You know, so why some of us are taller, some of us have blue eyes, some of us have green eyes, etc., etc. So it's those differences that explain why we look different, and to a large extent also explain why some of us get some cancers and other individuals get different kinds of cancers, or indeed heart disease or diabetes, in fact any common human condition. So here's the zygote at the point of conception. It starts dividing. So this single-celled zygote divides, undergoes DNA replication, becomes 2 cells, which then becomes 4, becomes 8, becomes 16, etc., etc., etc. And every time the cell divides, you get these errors in the DNA that are caused by DNA replication. And I put some figures on here. So in fact, every time a cell divides, you get about 120,000 mistakes. Sounds alarming, I know. But you've got to remember that most of the human genome is actually doesn't code for anything, it doesn't do anything. It's only about 3% of the human genome actually is important for doing its job, essentially. So the cell divides, the embryo develops in utero, inside its mummy's tummy, until eventually you get one of these. This is me as a little nipper when I was about 6 months old in 1970. And so even at this point in my life, I've just been born, I've actually already got changes in my DNA that I've acquired through DNA replication as I've been growing in my mum's womb. So then, of course, as I grow up and ultimately turn into this, which is what I look like now, through this process of living, I've also been exposed to lots of interesting exposures that might also cause damage to my DNA. And I've put some examples on here. So, for example, diesel particulate, we know damages DNA. Eating lots of red meat, and particularly if you overcook red meat, that causes damage to the DNA in your colon cells. And also I put here O2. Now this isn't the mobile network, I should point out, although I have used their logo. I'm hoping they don't sue me for that. But what I mean by this is molecular oxygen, so oxygen in the air that we breathe. So as a species, we use oxygen to generate energy. And in fact, it's an incredibly efficient way to generate energy that allows us to go out and go about our lives. But unfortunately, oxygen also damages DNA quite efficiently. And our cells have evolved very careful mechanisms to keep oxygen away from DNA, but they don't absolutely work perfectly. So as I grow old from this point up to this point, not only am I acquiring these changes or mutations in DNA because of cell division, I'm also acquiring changes in my DNA because of the exposures as I go through my life, and including these ones here. So I should point out that I haven't always been bald. For a very short period in the 1980s, I did have a full head of hair that I'm very proud of. So the point about this is that in addition to these changes or these differences in our DNA sequence that we're born with, these acquired errors or mutations are actually inevitable as we grow old, and we can't avoid them. And also importantly, each cell evolves differently. And it's these acquired changes that ultimately lead to cancer in humans. So just to sum up, there's two kinds of variation in DNA sequence. Those that are present from the point of fertilization in our genome, we call these constitutional variants. I've mentioned those already at the beginning. And it's those that, as I mentioned, affect our risk of developing all kinds of human diseases, including CLL. And in fact, we found over 40 of these variants in the human genome that confer risk of developing CLL. And one particularly is in a gene called IRF4, interferon regulatory factor 4, and this is probably one of the strongest ones that contributes to risk of developing CLL. But so far, as a community, we've discovered about 40 of these variants in the human genome. So we know they affect risk of developing CLL. So the reason that we've been asked to speak to you today is the paper that Dave and I have done has really been focused on trying to address whether these constitutional variants that exist in the human genome could affect disease progression. And I'll come back to that in a few minutes. So as well as these pre-existing alterations in DNA, I mentioned also these acquired alterations, these acquired errors or somatic mutations that we see in DNA. And some of these Dave's already mentioned. And these are age-associated or exposure-driven. And some of those include, for example, mutations in genes like p53 and ATM that some of you may have heard of with respect to CLL. And it's these ultimately that cause CLL or cause a B cell to become leukemic. And they also have a big impact on prognosis. So CLL with a p53 or an ATM mutation tends to have a worse prognosis compared to CLL without these mutations. So Dave and I, a number of years ago, decided that we wanted to see whether we could identify any constitutional variants, these pre-existing alterations in DNA that might be associated with disease progression. So in order to address this, we set up a UK-wide collaboration, and I put some of the centres involved in our study on this figure here. So you can see the great and the good of CLL research in the UK, and many of you will actually probably know some of these people, particularly if they work in a centre local to you. So what we've done here is we've collected data and DNA samples from 755 CLL cases. And these cases have a median follow-up of about 12 years. So of course, one of the issues with CLL is that, from a scientific point of view, is that the disease does progress, generally very slowly. So these sorts of studies are easier done retrospectively when you've already got lots of follow-up on these patients. So what we've done in our study is we've, using modern techniques, modern technology, we've been able to look at, in each of these 755 cases, we've been able to look at over 5 million of these pre-existing constitutional variations in the human genome, and really ask the question whether we can find any that associate with disease progression. And the answer is, of course, yes. So we found two. And I hope you can see this. So we found two markers in the human genome that associate with risk of progression, one on chromosome 6 and one on chromosome 10. And these numbers here, RS3778076, and this one here, these are what are called reference sequence numbers. So they each denote a single base position in the human genome out of 3.2 billion. these numbers refer to just 2 single positions in the human genome. So you'll see at this position on chromosome 6, the 377 position, humans either will have an A letter or they'll have a C letter. And in fact, we can see that most patients actually have the C variant, but there's a small number, about 6 to 7% of patients that have the A variant. And what this figure is, this is called a forest plot. And what we've done here is we've looked at patients with the A variant and compared their progression to those with the C variant to see whether they progress more quickly. And in fact, they do. And the risk of progression for patients with the A variant is almost twice as quick as those patients with the C variant. So this red line here indicates that progression is equal between the two variants. And what you can see is that anybody with an A, see all the data is pushed to the right. So patients with the A variant progress about twice as quick on average as patients with the C variant. And this chromosome 10 marker exactly the same. In this case, we've got a C or a T variant. And in this case, patients with the C variant progress quicker compared to patients with the T variant. So this variant is a little bit more common. Patients with the C variant represent probably about 10 to 12% of humans. So what does this mean? Well, Dave showed you some of these plots earlier where we can follow progression of CLL patients. What you can see here is that in the green line, we've got individuals who don't have either of these two risk variants. So in fact, they have the C variant at chromosome 6 and the T variant at chromosome 10. So they have the low risk version of the gene. You can see that progression is actually relatively slow. So this is from point of first diagnosis to the point of needing treatment. So patients who have one of the risk variants, you can see they progress a little bit quicker. And by about 20 years, about 75% of those patients have reached the point of treatment. In purple here, you can see patients who've got both of the risk variants at chromosome 6 and chromosome 10, and you can see that they progress again a little bit quicker. So at 10 years, about 85% of those patients have progressed to the point of needing treatment. So one of the really good things about these markers in the genome, these variations in the human genome, is that they're actually incredibly easy to test for. Costs a few pounds in our laboratory to test for these. To get them done commercially, you have to pay a little bit more because there are quality control issues that need dealing with. But we can test for these very easily. The technology is very straightforward and it can be done relatively cheaply. So in combination with other markers, and some of these I've already mentioned and Dave's mentioned as well, P53, which is the 17p mutation, or the chromosome 11 mutation, which affects the ATM gene. Also with things like CD38 surface expression that you might have heard about. In combination with these markers, we can now develop a more accurate model for predicting who's more likely to progress from their CLL and who might require treatment during their lifetime. So this is in fact the model that we've put together for this. So I've included here some of the markers that I've mentioned already. So CD38. So CLL that's positive for CD38 progresses about twice as quick to the point of needing treatment compared to CLL that is negative for CD38. Dave mentioned IGVH earlier, and having an unmutated IGVH is actually associated with a poorer outcome. And I've built in here the two markers that we've discovered in our study, chromosome 6 and chromosome 10. And you can see here how they relate compared to the other markers. So they're not quite as good or quite as powerful as IGVH, but they're approximately the same as CD38 in terms of their power. So why is this important? Well, Dave touched upon this already. Some of the clinical trial data that he mentioned. So this is an early report from the CLL12 trial that's been conducted by one of the German groups, who demonstrated that in patients with high risk markers, asymptomatic patients, but with high risk markers, like, for example, being CD38 positive. These patients, if treated early with ibrutinib, the B-cell receptor inhibitor ibrutinib, they progress to the point of needing treatment much slower. And in fact, they progress approximately 5 times slower. So, of course, with an accurate model for predicting or identifying high-risk patients, we can maybe better identify those patients who might benefit from earlier treatment of their CLL. So I think I'll stop there. Thank you very much for listening, and I'll hand you back to Brian.